Hello everyone. This video is for February 1st and 2nd. So my green day, on this day, you were in class. My red day, your work will be asynchronous, meaning that you will have to do this on your own. Hopefully this video will be very helpful either way. But of course, if there's any questions, please let me know as soon as possible. So I just have one announcement. And it's going to be about ISTEP. So by the time you see this, you probably already know about it. Also, February is Black History Month. So in honor of that, I want to take every day and introduce you to a Black scientist. So we have our Black scientist of the day. So today I want to start with a Punnett Bell work so that we are staying on top of our knowledge of Punnett squares, genotypes, phenotypes, all that fun stuff. I have a few slides where I want to review mitosis and meiosis. So we learned about both of those last week. Then I want to end with a Venn diagram assignment about mitosis and meiosis. So for my green days, the rest of the time in class was spent for working on any missing assignments, or there was an extra credit opportunity for you if you wanted that. For my red days, I'm also going to tell you about the extra credit opportunity, though since we didn't set out a block, this is just asynchronous. I'm going to tell you the things that you need to do. Of course, if there are any missing assignments, please still continue to work on those. If you have any questions, again, please let me know as soon as possible. So as you probably know, ISTEP is Tuesday, Wednesday. And this is for juniors and seniors. So freshmen and sophomores, you do not have to come to school those days. And we ask that you do not come to school those days. Pretty much every one of your teachers is going to be busy proctoring the ISTEP. So yeah. Work for all of your classes, like this one, is going to be asynchronous, meaning that you are expected to do this work on your own. I don't know what your other classes will be like, but this video will pretty much tell you everything that you have to do for Tuesday if you're watching this red days. If you're a green day watching this on Wednesday, I'll have another in case you missed it video for Wednesday, Thursday. So keep a lookout for those. But Tuesday, Wednesday will not come to school. Work will be asynchronous. I will still be close to my laptop, so if you have any questions, I'll try to respond via email or Canvas message as soon as possible. In the afternoons, I should be available to meet on Zoom if you have any questions that you would like me to help you through there. All right, so for our Black scientist of the day, today is Mae Jemison. So Mae was born in 1956. And in 1987, she became the first African American woman to be admitted into NASA's astronaut training program. And this was a huge deal because only 15 out of 2,000 were chosen. So this is awesome. She then became the first African American female astronaut in 1992. So 2 big milestones here. And as a cool little tidbit, she also has a medical degree. So not only is she a scientist and an astronaut, she's also a medical doctor, which is pretty cool. So currently, she is leading the 100-year Starship project, which makes sure that human space travel to another star is possible within the next 100 years. So pretty wonderful things coming from Miss Mae Jemison. All right, so the first thing we're going to do work-wise is the Punnett Bell work. On Canvas, you're going to go to your modules and all the way down to week 5. Again, week 5 has all that information in there and the agenda in case you'd like to take a look at it again. But we're going to start with Punnett Bell work, and this is today's date. So just like before, there's a problem and there's questions to answer. So in this Punnett square problem today, we have in cabbage butterflies, white wings are dominant to yellow wings. If a heterozygous butterfly is crossed with a homozygous recessive butterfly, what are the possible genotypes and phenotypes of the offspring and the percent chance for each? Like usual, the first few questions are going to be about whether you can tell me information that you read from the word problem. The first two are asking the dominant F allele, what phenotype does that refer to? So I'll tell you up here. What phenotype does a dominant F give us? And then answer the same thing for recessive Fs. The recessive allele, what phenotype will that give us? So that's just making sure you know how to read the problem. For number 3, you're going to copy and paste this table into the text box, and then you're going to fill in your Punnett square. So I'll kind of show you how to do that in a second. I tried to make this as least confusing as possible, which probably means that I made it somewhat confusing. So while the Punnett square is usually these 4 squares, I want to make sure I give you space to write your parents. So up here in these boxes, you're going to write your parent genotypes. So for example, we have a heterozygous and a homozygous recessive. So up here, I would have a big F in this one and a little f in this one for my heterozygous. And then, of course, these are going to have 2 letters, one from the top, one from the left, like our usual Punnett square. After you make that Punnett square, you're going to list out all the offspring's genotypes. So of these 4, B, C, D, and E, what genotypes do you see, and what percentage do you have of each? Then you're going to do the same thing with phenotypes, okay? Genotypes and phenotypes are different, so make sure that 4 and 5 are not the same answer because they should not be. All right, so if you just go to Submit Assignment, a text box will pop up. And what I mean by this table is you should just be able to copy and paste it. So copy and then paste. And it'll look like that, but all you have to do is just expand it a bit and you just type in your parents and then you just bring one from the top, bring one from the top, bring one from the left, like our usual Punnett squares. And don't forget to answer questions 1, 2, 4, and 5 as well. I'm looking for 4 written answers and a Punnett square. So then you'll just press Submit Assignment, and that's it. So we did not go over the answers to this one in class. So if you have any questions about it in order to get it done, please let me know. At this point, you should be pretty comfortable with this. It should be like second nature. If it's still not there, let me know. We need to get you there. After you've done the bell work, I want to go ahead and review mitosis and meiosis real quick so we are all on the same page about the information. So before I start mitosis, meiosis, I want to talk about the cell cycle. So this was briefly talked about in the Edpuzzle video that I gave you last week. But the cell cycle is essentially the life cycle of the cell. So as the cell progresses through its life, what happens? So I have a picture up here of the cell cycle. It's split up into these different stages, but we really haven't talked about like what happens in G1, S, or G2. We just kind of collectively call this interphase. So what's important to know about interphase is that during this time, the cell is doing its normal thing. It's functioning the way it needs to function to make sure that our bodies work the way that they do. What else is happening during this phase is the cell is preparing for division. So what needs to happen in order for the cell to divide? The DNA has to double or copy itself, so each cell has its own copy of the DNA. And during interphase, we also have checkpoints, and that's kind of what these are. These little stops or steps between the stages. And what's happening here is we're just checking in with the cell and making sure that it works properly, that it's good to go. If there's any mistakes in the cell, we don't want to make copies of bad cells. So we have to make sure it's good. It's a good cell to replicate. The cells spend most of their life in interphase. So here's the whole cell cycle. You can tell that about 90% is interphase. The rest is this M phase, which we know as mitosis. So after interphase, it then goes through mitosis where it divides. So for mitosis, mitosis is one of the two types of cell division processes that we talk about. This one specifically happens in all of your body cells. We also call them somatic cells. And these are literally all your body cells, your blood cells, bone cells, muscle cells, skin cells, everything except for the sperm and egg. The cells that are made from mitosis are diploids, meaning that they have two copies of chromosomes. So all of our cells have one copy from mom and one copy from dad that make us who we are. Mitosis also only has one division. We take one cell, we split it in half, essentially. So just once, we have one division. And then at the end of it all, we have two identical cells. It's like we copied and pasted. So they're the exact same. So this is the summary for mitosis. If you have this down, you have quite a bit of information down, and you're good. So meiosis is very similar but different. So this is another type of cell division as well, but this one happens in your sex cells, so your sperm or your egg. These cells end with haploids, or this division ends with haploids, which means that they just have one copy of the chromosomes. So think about what it takes to make a baby. We have a sperm and an egg that come together, and we have 46 chromosomes in our body cells. So we have to make sure that our end product is 46 chromosomes. Otherwise, we get some outrageous number, and it might not work. So our gametes have to each have 23 chromosomes so that when they come together, sperm and egg come together, 23 and 23 make 46 chromosomes, and we make a good human. In meiosis, we actually have two divisions. So we have meiosis 1, where you take one cell and you split it up into two. And then we have meiosis 2, which takes those two cells and splits them even further. So we have two total divisions here. At the end of it all, we have 4 cells that are genetically different. Okay, that's a big point. They are not identical to the first one. They are genetically different. And that is because of this process called crossing over that happens in meiosis 1. We'll talk a little bit more about that later. But all of these are genetically different. That is why we are very different from our family in appearance. So while we're very similar, we might have the same eye color, hair color, dimples. We might look the same from our parents or our siblings, but we're still very unique. And that's because of these 4 genetically different cells that are created by meiosis. So once we get together and make babies, it's kind of a toss-up as to which types of genes they might get from you and your partner. So that is my quick summary of the cell cycle, mitosis, and meiosis. If you understand those bits of information, then you are on the right track. So hopefully that helped clear things up between those recap notes, the meiosis notes I gave you last week, and the mitosis Edpuzzle / video recap. You have a lot of information that I've given you. Now you're going to take that information and you're going to show me what you've learned. So going back to our modules on Canvas, our next assignment is mitosis versus meiosis Venn diagram. So here you're going to click here to make a copy of the document. And the first slide gives you the directions already. So you're going to click and drag each statement that's on the right to its correct area on the Venn diagram. So we're looking at mitosis versus meiosis. So here we are. So these are the statements that I have for you. All of them, you can click and drag them to the correct spot. So you'll just read them, like DNA divides through PMAT, so prophase, metaphase, anaphase, and telophase. So then you'll tell me, does that happen in just mitosis? Does that happen in just meiosis? Or does that happen in both? And then you'll put them in the right spot. And all of these have a correct spot on the Venn diagram. This is not a quiz. Feel free to use your notes. And if you have any questions on it, just let me know. That's pretty much it. When you're done putting all these statements in the correct spaces, you're going to go back here, click on the Submit Assignment button. You're going to use the Google Drive tab to find your copy of mitosis versus meiosis Venn diagram and turn it in. And bam, that's it. If you have all of your assignments turned in, especially that Venn diagram that we just did, if you would like, I also offered an extra credit opportunity. So this phases of mitosis versus meiosis extra is that extra credit assignment. So this is completely optional. You don't have to do this, and I try to make sure it's in multiple places so you don't do it on accident. But I will happily take this if you complete it on time and if you do the Venn diagram first. So you have to do the regular credit before getting the extra credit. So for this one, if you choose to do it, you'll download this document to your computer. So you'll click on that twice, make sure it downloads. And it looks like this. And then you're going to upload it into ICYMI. So that's what it says in the next few directions. So you're going to open it up in ICYMI. And then you're going to use the drawing tool. So here are the two phases that we've been talking about. If you want to do this extra credit, we're going to take it a little step further, and you're going to draw what happens in each of these stages. So in mitosis, what does prophase, metaphase, anaphase, and telophase look like? In meiosis, what does prophase, metaphase, anaphase, telophase 1 and meiosis 2 look like? So what's happening in the cell here? So when you're done, you're going to download that back to your computer using those annotations and turn it in here. And again, that is completely optional if you would like to do it.